Good morning, students. In this class, we are going to learn hidden Markov models. We have studied phylogenetic tree and parsimony. The relationship of sequences are considered for these concepts for understanding. The number of mutations or insertions that occur for this kind of evolutionary changes are studied or considered when we study phylogenetics. We know that sequence information is increasingly abundant. Markov process is a probability based approach or model. To understand Markov process, consider the throw of a die. We know that whenever we throw a die, different value or same value can occur, and with different time different types of results occurs. Markov process is the one where you have a physical system that stepwise goes through some kind of change. We know this die will get, when we throw, different values in different time. Whenever we get one value, during the next throw we expect another value. But we know that the each throw is independent. It does not depend upon the previous action. In Markov process, the change is the transition from one value to the next. A Markov process is characterized by the property that the change is dependent only on the current state. The history of previous states is immaterial. HMM, hidden Markov model, is called hidden because only the symbols emitted by the system are observable, not the underlying random walk between states. As mentioned earlier, a Markov process is a probability based approach or a stochastic model. It can exist in various states and emits a symbol each time you visit it. For example, the different die values. Each state can connect to other states or maybe itself. That is, a die, if once throws, the value can be 3. Again, if the die throws the value again, it can be 3. So each state can connect to other states or to itself with the probabilities attached to each transition. The sum of all transition probabilities is equal to 1. It is a universal thing. The total probability of all the occurrence of events will be equal to 1. So in this figure, this is shown. This physical system can have 3 states, A, B and C. The system can exist in these 3 states, A, B, C. Each time when it occurs, the next time there will be another state or it can be the same state. The probability of the movement from A to B, the state is given as 0.3. The probability from A to C, the probability is given as 0.1. The probability to occur the A itself again is given as 0.6. And when we add the sum of all transition, the probability will be 0.6 plus 0.3 plus 0.1 is equal to 1. This is Markov process probability model. Now let us think how this probability model can be applied in biological context. In biological context, we have sequences of either DNA or sequences of protein of amino acids. We can have a statistical record of all the occurrence of various states from the sequence. We know, or it is understood, that there is a hidden process that generates a sequence of amino acid residues where chance, that is based on specific probabilities, play an important role in determining the exact sequence being produced. Now, for modeling biological sequences, we have to follow these steps. Step 1 is we need to collect the set of sequences of our interest. Then step 2 is we have to define the vocabulary V is equal to x1, x2, x3, etc. xn. For example, in the case of DNA sequence, the various vocabularies are adenine, thymine, cytosine, and guanine. So the vocabulary V x1, x2, x3, etc. xn is V is equal to set of A, T, C, G, and we know we have four kinds of vocabularies. So N, the number of sequences with this vocabulary is A, T, C, G is N here. N is equal to 4. Now, based on this, we need to develop a model, and this model is then used for generating a typical sequences from the class of training sequences. An hidden Markov model can be visualized as a finite state machine. Finite state machines move through a series of states and produce some kind of output either when the machine has reached a particular state or when it is moving from state to state. The hidden Markov model, working as a finite state machine, generates a protein sequence by emitting amino acids as it progresses through a series of states. Each state has a table of amino acid emission probabilities similar to those described in a profile model. There are also transition probabilities for moving from state to state. The things will be more clear when we see the next slide. This figure shows the Markov model for DNA sequence. The arrows show the various transitions in the model. That is, after the sequence letter A, the next symbol of transition can be either A itself, or next state can be G, or next state can be T, or next state can be C. So all the transitions from all the symbols are shown in this Markov model for DNA sequence. Suppose sequence of events or DNA sequence is CGGT. The probability of occurrence of the sequence CGGT can be expressed with Bayes' theorem. That is, probability of CGGT is equal to probability of occurrence of C into probability of occurrence of G in such a way that C has occurred previously into probability of occurrence of G in such a way that G has occurred previously into probability of occurrence of T in such a way that previous occurrence is G. When we make Markov model, the Markov model of the DNA sequence can be modified with the beginning information for the sequence of events CGGT. So from the beginning, the arrows of various transitions in this model to all the symbols are shown as the modification of the previous Markov model for DNA sequence. Markov chain models can also have an end state. The end state can be used to represent a distribution over sequences of different lengths and also for giving preferences for ending sequences with certain symbols. This is shown in this figure. A linear hidden Markov model for a family of nucleotide or amino acid sequences is a set of positions that relate to columns in a multiple sequence alignment. Each position will have three states: matching state, insert state, and delete state. Probabilities or cost, that is negative log probabilities, are associated with each character emission and each transition between states. The alignment of a sequence is the highest probability or lowest cost path through the hidden Markov model. If you are given a multiple sequence alignment of a particular domain family, you can use statistical methods to build a specific hidden Markov model for that domain family. The relevant probabilities required are estimated from the frequencies in the alignment together with other data. You can then set up this hidden Markov model to test other sequences whether they match this domain family or not. Hidden Markov model can be set up so that the typical operations like insertions, deletions, and substitutions can be handled. The model of the hidden Markov model determines which probabilities need to be estimated and what kind of matches are allowed. Hidden Markov models are now used for sequence modeling, multiple alignment, and profiling. Hidden Markov models offer a more systematic approach to estimate model parameters. Like an ordinary profile, it is built by analyzing the distribution of amino acids in a training set of related proteins. However, the hidden Markov model is a dynamic kind of statistical profile and has a very complex topology than a profile. The primary advantage of hidden Markov model over profile is that they can be automatically estimated or trained from unaligned sequences. Now let us understand the components of profile hidden Markov model. A profile can be thought of as a series of amino acid probability distributions, one for each M state as shown in this figure. Profile hidden Markov model use position specific scoring. Unlike traditional pairwise alignment algorithms such as BLAST, FASTA, and Smith-Waterman, hidden Markov model use position-specific scoring for the matching or substitution of a residue and for the opening or extension of a gap. Position-specific scoring allows hidden Markov models to characterize entire families of sequences by modeling the extent to which the regions should be conserved in a multiple alignment. The probability of a gap or insertion is position specific in this model. In this figure, each match and insertion box has a probability distribution of amino acids. The transition probabilities from each box add up to 1. Each delete state adds a delete character to the sequence with probability 1. To put it in the standard notation, M is equal to match state output, that is output. I is equal to insertion state, that is output. And D is equal to delete state, that is no output. Graphical representation of a typical profile hidden Markov model is shown in this figure. If a box contains a uniform distribution across the 20 amino acids, then the probability of any amino acid is 0.05. If all the transition probabilities are equally probable, then they are each of 0.33. Using these average values of 0.33 for the transition values and 0.05 for the probability of each amino acid in each box, then the probability of a sequence occurring is the product of all the transition probabilities in the path and the probability that each stage will produce the corresponding amino acid in the sequences, or that 0.05 into 0.033 equal to 4 into 10 power minus 10. The secret of hidden Markov model is to change these values of 0.05 and 0.033 at each step in the model by training the model with a set of sequences. Training involves finding all of the possible paths through the model for each sequence and then leaving a memory of the path by adjusting the distributions and transition probabilities. For multiple sequence alignments, you need 20 to 100 sequences to train a model. The model is also improved by using Bayesian prior distributions in the match boxes. In addition to the start and end states, there can be 3 other classes of states, that is main, delete, and insert. The following is a representation of an hidden Markov model process, that is S is equal to start M1 etc. Mn, I1 etc. In plus 1, D1 etc. Dn, and end. So here capital N is the length of the model, that is average length of sequences in the family. Main and insert states always emit an amino acid symbol. delete states are mute. The self loop on insert states allows multiple insertions. So now let us discuss the building of an hidden Markov model. One of the important issues is how to create a hidden Markov model in the first place when given a particular set of related training sequences. There are two cases for multiple alignments: ungapped multiple alignments and gapped multiple alignments that imply that there are deletions in the alignments. For the ungapped case, assume sequence length is n. We can build the model as shown in this figure. For ungapped alignment with hidden Markov model. For each state, the transition probabilities are 1 because we have to traverse all the states. As for emission probabilities, each state has a list of probabilities for emitting different characters. For the gapped case, one needs to consider deletions in the multiple alignments and the model will be a little different. For the gapped case, the profile hidden Markov model can be constructed from multiple sequences as shown in this figure. Here in step 1, we make the 4 matching states based on the starting sequence. The multiple sequences are shown as model based on this sequence. Then we make the 4 match states on this sequence. Then the probability of amino acid distribution is shown with each matchbox. In the second step, we add insertion states where there are insertions, that is represented on the left side sequence in small letters. So insertion states are written. Now in step 3, we add deletion states. The deletion states are marked with star in the sequence. Now after adding the insertion and deletion states, the graphical representation of profile hidden Markov model will look like this. And finally, the profile for each state can be represented with the weights W1, W2, etc. for each of this sequence based on the count of the frequency of each amino acid scaled by the sequence weights W. Once the hidden Markov model has been constructed, we need to score the hidden Markov model and arrive at the alignment. But there are 3 basic problems for hidden Markov model that need to be addressed. One problem is model structure learning. Second problem is evaluation problem. Third problem is model training. Model structure learning is that suppose we have given the observation sequence and the model, how do we choose a corresponding state sequence that is optimal in some meaningful sense? This problem is called model structure learning. evaluation problem means we have given the observation sequence and the model. How do we efficiently compute the probability of the observation sequence? This is our evaluation problem. And model training is how do we adjust the model parameters to maximize or to get the optimum position? That is model training. There are three main algorithms for solving these problems. Viterbi algorithm is used to solve these problems, which is based on dynamic programming. Then forward algorithm can be used, and third, Baumwell algorithm can be used. Now let us discuss these algorithms briefly here. Viterbi algorithm. Viterbi algorithm is used to produce multiple alignments of a set of sequences. The probability or the score can be calculated for all possible alignments to a model, and thus the most probable multiple sequence alignment can be found. Viterbi algorithm is a dynamic programming technique that can find the best alignment and its probability without going through all the possible alignments. For each of the sequences, the alignment to the model is found. The columns of the multiple alignments are then determined by the match states. All the amino acids matched to a particular match state are placed under each other to form the columns of the alignments. For sequences that do not have a match to a certain match state, a gap character is added. The model consists of a linear sequence of nodes with a begin state and an end state. Each node between the beginning and end state corresponds to a column in a multiple alignment. Hidden Markov models are similar to profiles in this respect. Each node in an hidden Markov model has a match state, insert state, and delete state with position specific probabilities for transitioning into each of these states from the previous node. In addition to a transition probability, the match state also has position specific probabilities for seeing, or more formally emitting, a particular residue. Likewise, the insert state has probabilities for inserting a residue at the position given by the node. There is also a chance that no residue is associated with a node. That probability is indicated by the probability of transitioning to the delete state. Both transition and emission probabilities can be generated from a multiple alignment of a family of sequences. A hidden Markov model can be compared, that is aligned, with a new sequence to determine the probability that the sequence belongs to the modeled family. The most probable path through the hidden Markov model, that is which transitions were taken and which residues were emitted at match and insert state, is taken to generate a sequence similar to the new sequence determines the similarity score. So now let us define the Viterbi algorithm stepwise. The Viterbi algorithm can be defined as a matrix where the columns of the matrix are indexed by the states in the model and the rows are indexed by the sequences. Deletion states are not shown since by the definition they have zero probability of emitting amino acid. The elements of the matrix are initialized to 0 and then computed with the steps. Now the steps are: step 1, the probability that the amino acid A was generated by state I0 is computed and entered as the first element of the matrix. Step 2, the probabilities that C is emitted in state M1, that is multiplied by the probability of the most likely transition to state M1 from state I0, and in state I1, that is multiplied by the most likely transition to state I1 from state I0, are entered into the matrix element indexed by C and I1 by M1. Step 3, the maximum probability, that is maximum of I1, M1 is calculated. Then step 4, a pointer is set from the winner back to state I0. Step 5, repeating the steps 2, 3 and 4 until the matrix is filled. Forward algorithm is used to calculate the sum over all paths inductively. The forward algorithm is similar to Viterbi. However, in step 3 of Viterbi algorithm, a sum rather than a maximum is computed and no back pointers are needed. The probability of the sequence is found by summing the probabilities in the last column.